Again, it's a pleasure to introduce the next symposium speaker, Elina Niño. And we've been very fortunate in entomology and nematology here. We've actually hired 4 new scientists over the past year, so it's quite remarkable. Elina is one of our newer members in the department. She's been here for less than a year. And a little bit of her background, basically, she has a BS degree in animal science from Cornell University, and then down to NC State, where she worked with Christina Grosinger when she was there, has a master's degree in beekeeping, entomology, beekeeping, and then she followed Christina to Penn State, where she received her PhD. She also did a postdoc with Christina, and basically has come on board here as our Cooperative Extension Specialist in apiculture. And so in that context, she has a 70% appointment in Cooperative Extension and a 30% research appointment. And she has huge shoes to fill, because obviously she has come in to try and take over the responsibilities of Eric Musson, which is almost impossible to do. And again, I'll just say one more thing. Obviously, you are all stakeholders, right? So in a sense, she is really supposed to be responsive to your needs. And probably every state has stakeholders, but in California, we carry that to an extreme. We obviously have more bee colonies here. We have more industries that rely on pollination services. We have the largest queen rearing industry in the United States, and we probably have more urban individuals dealing with beekeeping than in any other state. So in that context, to say that she has her hands full is an understatement. Her research mainly focuses on queen health and queen rearing, and I think she'll talk a little bit about that. She has other applied work going on, and she is also starting to begin the preparation for a master program in beekeeping, and I'm hoping she talks a little bit about that, and maybe some of you will be, and hopefully many of you will be interested in that. So, and I want you one more thing. We actually hired a team, so not only do we have Elina, but we have Bernardo, her husband, as well. So we welcome both of them in terms of to our beekeeping group here at Davis. So with that, Elina.
Thank you, Michael. That was absolutely no pressure whatsoever. Thank you all for being here. I want to start off by saying thank you to the Robert Mondavi Institute, the Honey and Pollination Center, especially Amina and Claire. They've done a wonderful job, obviously, in organizing this symposium. And, of course, the Kaiser Foundation for their support at the Department of Entomology and Nematology. I am Elina Niño, the new extension specialist in apiculture, and I've been hearing a lot about filling Eric's shoes, and I always come back with saying, well, at least I'm the younger, better-looking version of Eric. So, Eric, if you're there, I know you're here. That's how it is. So I've been asked by Amina to talk a little bit about the best management practices to supporting honeybee health. Of course, you've heard a lot about honeybees this morning, as well as the native pollinators, but I am an apiculturist, so I will talk mostly about honeybees. And I get asked all the time by beekeepers and others who are not beekeepers, how are the honeybees doing these days? Well, I'm sure you all have heard about this survey that's being done by the Best Informed Partnership. It's been done by for the past 8 years. This is the latest full set of data that they have from 2013, 2014. The year has been looking pretty much better than the year before, a little bit worse than the year before that. So you can see there's that sort of not a very consistent pattern, but the point is we're still losing honeybee colonies. And I'm really excited to see what they come up with. I know the preliminary data might be coming out in May. They've been pretty good about pushing that data out, so I'm really curious to see what's happened this year. But again, the point is we are losing honeybee colonies still. Okay. You've heard a lot about this. What is affecting the honeybees? There are a lot of things. It's a complex issue. It's not a simple thing. It's not one answer fits all questions, right? It's not one solution fits all the problems. So they are affected by many parasites, many pathogens, viruses included bacteria, nosema, many of them, well, I shouldn't say many of them. Almost a lot of the colonies have mites, so I was really surprised to hear from Aid Mead when she said that half of her samples basically didn't have the rot, so that's looking promising. Of course, nutritional stress is a big issue, and of course everybody asks me about pesticides and how we're dealing with pesticides, but particularly neonicotinoids. This is why I'm really nervous about giving this talk, because inevitably somebody gets mad at me. So hopefully I'm here to tell you what is available for you, and what you are going to do is really up to you. Okay, so this is the outline of the talk. I'll be talking about the best management practices for the beekeepers. Actually, so now you know about me, you've heard from Michael. I want to get to know you a little bit. I will be biased, so I'm going to ask you, how many of you are beekeepers? Pretty good. I like that. So you guys can keep me in check. Then I'll talk a little bit about the best management practices in agriculture, and then a little bit about what anybody can do, regardless of whether they're a grower, a beekeeper, what anybody can really do to support the honeybees and pollinators in general. And I will take this opportunity to tell you just a little bit about what we're doing at the honeybee lab here, Niño Honeybee Lab here at UC Davis. We're still a pretty new lab. We've been here 9 months, so I really want to spread the word. And I promise next year when I come talk to your beekeeping association, I won't be talking about that anymore. Okay, so for the beekeeper, basically, what I really want to stress out is that it's really important for you to know what is going on in your colonies. I've talked to beekeepers. How often do they check their colonies? Of course, this is going to depend quite a bit on the size of your operation. How many of you have fewer than 10 colonies? Okay, so you might be able to go into those colonies a little bit more often, pay more attention to every individual colony. How many of you have more than, let's say, 1,000 colonies? So you guys might have a little bit more of a challenge to go and check out into each of the colonies what is exactly going on, but you still should be able to infer from just checking a few colonies, if you can't go to 1,000 or more, checking a few colonies so you should be able to know what is going on in your apiaries from there, because knowledge is really the power. You should also be sure to know the host and pest pathogen biology and the interactions. I've heard of, from several new beekeepers, they would come to us and ask us questions about, like, hey, I just got these new bees, what about, this? Like, wait a second, did you take a beekeeping course? Did you read some books? I think it's really important to familiarize yourself with what you're getting yourself into, because it's going to set you up for success as well. So there are many beekeeping associations who have wonderful courses. Of course, we're giving courses as well. So I would highly recommend you, start with good books. We have a lot of them. We've given some of them away. And definitely go visit some courses. If you do the courses, often they will talk about various symptoms of specific stressors. So we saw the honeybees have to deal with pathogens, honeybees have to deal with parasites. So what are some of the symptoms? What are the things that you should be looking for in your colonies to be able to appropriately take care of those colonies? And then, as always, as Marla mentioned, IPM has sort of fallen by the wayside, but prevention is always better than intervention. You should definitely use some sort of an integrated pest management program, integrated pest management approach, which is basically decision-making process based on understanding the host and pest biology, host-pest interaction, and it's action based on thresholds. So again, as I said, you should know what's going on in your colonies, use that information to be able to best address the issues that you might be facing. Okay, so I'd really like to show this pyramid of IPM tactics, sort of a classical pyramid of IPM tactics. But what Penn State researchers have done, I think this is actually done by Miriam Fraser, at Penn State, she sort of modified it specifically to the beekeeping community. So in addition to to the general terms here, if you go up, you can actually see what would be more specific for the beekeepers. And I would like to cover some of these topics today, starting with, well, first of all, let's talk about some of the potential problems in the apiaries, right? We need to know what's going on. I've been talking about that. So, varroa mites, they're a biggie. I've been doing sort of polls of beekeepers, unofficial polls of beekeepers. The question that I ask often is, what do you see as the biggest challenge in your operation? What do you see as the number one problem? What do you see as something that we would be able to address to support you? And almost in all cases, they will say varroa mites. And from the survey that Bee Informed Partnership has been doing, and other researchers in Europe as well as in Canada, who've been seeing that the high infestation of varroa almost inevitably leads to colony loss. So again, varroa is a pretty big issue. Diagnosis, there are several things you can do. Sometimes you can use the uncapping fork that you use for honey extraction to uncap, pull the drones out, see how many mites are on the drones. You can use ether rolls, sugar shakes, sticky boards, and all of this thresholds are always a sensitive thing, depending on where you look. Sort of, I think this number has been milling about for a long time. If you do a sugar shake, for example, if you find 3 to 4 mites in that sugar shake, you should be doing something right away to start and control the varroa mite or manage the varroa mites. This one, again, is a kind of iffy, the sticky board, 5 to 10 mites. We've had, so Billy's the technician in my technician in the lab and also the facility manager. He's done a sticky board for several colonies out by the Layla facility. And I know that he's found some that have even, I think it's a 24 or 48-hour count. He's found up to 50 or so mites on that sticky board. And of course, all of us are thinking, oh, that colony is doomed. And guess what? They're not. They're actually still doing okay. So that's a little bit trickier. That's another thing that you should be keeping in mind, sort of maybe genetics of the bees. But one big problem that varroa is posing, as you've heard before from Amy and others, is that, okay, so varroa is feeding on the hemolymph, it's suppressing the immunity of the honeybees, but it also transmits all of these, transmits several viruses. And the honeybees have to deal with over 20 different viruses. It doesn't mean that all of them are transmitted by varroa, but certainly some of them are. So definitely keep that in mind when you're treating this. Unfortunately, when we're talking about diagnosis for the presence of the different viruses, it's not always so clear-cut. So we know about deformed wing virus, right? Deformed wing virus is pretty clear-cut. When you see this, most likely your bees have deformed wing virus. This, I wish I could play for you. It's a video. It was a bee that was treated with the Israeli acute paralysis virus. And it's really very sad. It's a paralytic virus, so hopefully you will not be seeing that in your colonies. But the point is, diagnosis is trickier than if you just had a mite that you can see. And then what's even more tricky is the threshold. Another issue is there are really no good treatments for viruses, right? There have been some RNAi technology potentially is being developed for the control of viruses, but it's definitely good to know what's going on in your colonies. And again, if you see varroa mites, chances are you might be seeing these viruses as well. Another biggie, and again, these are not the only things that you will find in the hive that the honeybees have to deal with. These are just a few things that I picked out that might be more serious or prevalent. Hopefully not American foulbrood, but it is a biggie. I have heard that beekeepers do have it every once in a while. It pops up in the state of California. So American foulbrood diagnosis is really pretty sort of straightforward. The larvae, what they do is they basically melt down. And what you can do, you can take a toothpick, stick your toothpick into the cell that you think has an AFB. You sort of twirl it around, stretch it, and if it stretches for several inches or more, you most likely have AFB. And then you can also look at some of the concave cappings here. They have black scales that they form at the bottom of the cells later in the later in the infection. And the threshold is basically any sign should require immediate action. Another one that's been around is Nosema apis. More so recently, it's been replaced by Nosema ceranae. It causes this, well, Nosema apis at least causes this bee diarrhea, what we call. Cerana seems to be a little less symptomatic. To diagnose it, we actually had a queen-rearing course where Ben Salmon, who's going to be talking a little bit later, and Rob Snyder came from the tech team, Northern California tech team, and they gave a really wonderful presentation about how you can diagnose Nosema. Of course, how many of you have microscopes at home? So not too many of you, but inevitably somebody raises their hand and is like, hey, I have a microscope. So you do need a microscope, a pretty powerful microscope. So if you do have a microscope, you would be able to probably diagnose that you have the Nosema. Another problem is we really don't know what the real threshold is for treatment. We know 1 million spores or more seems to be what's been floated around, but again, other factors can play a role in this. So for example, I know work by Steve Shepard and also Jeff Pettis have found that if the bees are exposed to fungicides, for example, they tend to succumb to Nosema more readily. Okay, so let's go back to our IPM pyramid. So we'll start from the bottom, which is really where you should be starting anyway. We should start with the prevention and some of the less controversial techniques, let's say, for treatment of these various diseases. One thing that you should always be doing, just like with your house, right? If you have children, you don't want germs milling about, you want to minimize the transfer of pathogens and also pests. So sanitation is a big one. Just basically practice good hygiene while you're working. A lot of the times I'll recommend to use different equipment maybe. If you have different apiaries, especially if they're sort of far apart, you can do that. Or an alternative, you can clean your equipment. 10% bleach usually works really well. Wash your protective clothing as often as you can. I know it's really a badge of honor to see a beekeeper white suit that's no longer white, it's more closer to black, but you might want to actually clean that because it could be transferring something you don't want in all of your apiaries. If you are using equipment, this is another thing that I like to mention to everybody is, well, the equipment can harbor a lot of things. So pesticides, you heard somebody was talking earlier, a lot of the pesticides will accumulate in wax, for example. A lot of these pathogens can stick around in the equipment. So the scales of AFB, you saw those little black scales. If you're not familiar with it, you might not be able to recognize that when you look at the equipment. So one of the things that I always recommend is saying, like, hey, be polite, say no thank you, or if you know the beekeeper, then it's fine. But if you really don't know the history of the equipment, it might be worse for you to get it for free than actually get something you don't want on that equipment. And as I mentioned again, don't use it if you know that there's a known incidence of American foulroot, for example. You can always wash the equipment. You could scrape the wax off of the equipment with the frames, for example, which would get rid of some of the pesticides. There are some other options that people have been talking about. Heat treatment, for example, can kill nosema, UV light, and I also already mentioned compound or home scraping, freezing frames. Amy already talked a little bit about gamma radiation. Not so useful as pesticides, necessarily more targeted towards the pathogens. So there are some options out there, but of course anything that might be really useful tends to be complicated and expensive, so might as well buy new equipment, right? Okay, pesticides, a biggie, right? Everybody talks about pesticide exposure. We'll hear more about that this afternoon as well. This is not easy. I mean, there are pesticides that are found in pollen, nectar, and also in wax of the honeybee hives. Many of them have been identified in there. So they are obviously collecting these pesticides. They're bringing them in. You can try and minimize the exposure as much as you can. This can be a challenge, especially in the agricultural settings, but even in the urban settings. And again, there are pesticides that the beekeepers use themselves. So as I mentioned before, you want to use that prevention rather than intervention. And there are some other options other than pesticides that you can apply for hives. And when it comes to urban settings, somebody posed this question. It's really difficult to regulate what the homeowners are doing in their backyard. And you're absolutely right. There's really no real measurement of how much is being used. People often think more is better. That's not necessarily the truth. So there are definitely challenges, and it could be challenging to minimize the exposure of honeybees or other bees to pesticides. But if you can do it, and we've heard a lot about the balanced nutrition, both from Marla, from Amy. Very important. Honeybees do need a diversity of pollen sources. Try and provide that for them. Again, a challenge. But if you have that opportunity, it'll be definitely beneficial for your honeybees to do that. You can place them in an area with a variety of floral sources. Again, I talked to somebody earlier. They were mentioning how it's really a challenge. We've reached sort of the honeybee hive capacity in the United States, basically. We're running out of food sources for the honeybees, so it's a challenge. But if you can provide this for your honeybees, I know they will be very grateful to you. We'll talk about that a little bit later when I talk about what everybody else can do to help honeybees, right? And then what the beekeepers often have to do here, especially in California, because we are facing the drought, is actually feed supplemental forage or feed supplements to the honeybees. This is often in forms of sugar syrup, pro-sweet, some pollen supplements, and I will never forget a new beekeeper asking me, my honeybees died in the middle of the summer. What happened? Well, did you feed them? Did they have clean, food access? Well, no. Did you provide water for them? Well, no. Do you know they have water around? I don't know. Well, maybe I think I would recommend first, try and support your honeybees as best as you can. And of course, that again might be difficult, but there are some supplements there, pollen substitutes that you can use. And a lot of the beekeepers, I came from Pennsylvania. Pennsylvania, we had the opposite problem. Everything was wet all the time, which is not great, but that did provide great forage. So I'm a little bit worried about this summer here, the first summer here. But I'm definitely planning on probably providing some supplemental food for the honeybees. So that would be an option for you if you are interested in doing that. And I mentioned this already, but please do provide a clean water source for your bees. And that could be something like I just heard somebody mention, basically dig a hole in front or next to your hives and pour some water in there, pour some clay in there. As long as they don't drown, you can do something simple like a bird bath, for example. Fill it in with pebbles, add some clean water into it, refill it as needed. Just so your bees can have something that they can depend on, and they will go to it and really appreciate it. Another thing that you can do, and I really like this option because this is really most of my background, is studying honeybee queen reproductive and mating behavior and factors that are affecting the mating and reproduction. So I'm really interested in potentially breeding. And one of the things Marla mentioned actually is you could potentially get hygienic honeybee stocks and replace your queens. Put the hygienic stocks in your colonies, which then is usually can take care of varroa and some other diseases as well. There are several stocks that are available for purchase, and here in the state of California, there's a lot of interest for breeding your own stock. So even if it's not necessarily hygienic honeybee stock, there's a lot of push and a lot of interest for breeding local bees. And those local bees, the idea is that they would be more adapted to the local environment, so actually they might be better off dealing with some of the parasites and pathogens that they're exposed to as well. Then we have physical mechanical control. This is mostly for varroa mite control that I'm talking about here. A lot of them you can see a lot of them here. A lot of them you've probably used or at least heard about, and the best is probably to use sort of a combination of all. Now this might be a little more applicable to those who have fewer colonies as well. So that's definitely potentially an option, especially if you don't want to add more miticides into your colonies. Now let's talk about pesticides. We have sort of biorational pesticides, what we call sort of softer options. Some of these are formic acid. A lot of essential oils are being either used or are being tested for further use. And then, of course, we have the conventional pesticides that Brian already talked about, pyrethroids, organophosphates, Niño, other types of pesticides that might not be so great. Okay. So this is just a list for those of you. I see a lot of people snapping photos up there. So this is just a list for you that you can take back home and look at it and say, hey, these are some of the options that I might be able to use for, varroa mites, for nosema, for the American foul brood, or European foul brood as well. Some of these, keep in mind, have cautionary statements coming with them, and these are my cautionary statements. You won't find these on the label. Some of them have shown to have fruit toxicity or negative effects on queens. With Coumaphos and fluvalinate, the resistance has definitely been recorded in varroa mites. There's Amitraz. It's been approved for use in the state of California fairly recently. The resistance has not been recorded here yet, as far as I know, but I know it's been recorded in other countries where it's been used for a little while now. And this is, of course, I'm sure, I beg you, who's seen this graph before? Okay, a few of you. I was hoping all of you have seen it before. This is from a paper written by Chris Mullen, Marianne Fraser, Jim Fraser team. These are the pesticides that they have found in the colony matrix back in 2010. This was published, I think. And one of the two things that they found was fluvalinate and Coumaphos. And if I go back, Coumaphos and fluvalinate, right? So those are the pesticides that the beekeepers themselves are using, are pretty prevalent there. So as a cautionary thing, always follow the label instructions, use the correct dosage. I already mentioned, people think more is better, not necessarily, not always, so keep that in mind. Look at the colony strength. As a novice beekeeper, we've applied formic acid once. The pad of formic acid in Pennsylvania, we started off like, okay, we got our new colonies, got new packages, we'll throw in that formic acid, get rid of the mites as soon as we can. Of course, the whole colony died because it was really weak. We were just, foolish, right? So we all make mistakes, but definitely think about the colony strength when you're applying these things. Appropriate time of the season. A lot of them come with appropriate temperature that you have to have outside temperature to apply these. And check to see if your treatment is working. If it's not working, then you shouldn't keep applying and try something else, right? Okay. So, agriculture. There are several places where you can look up some of these cautionary or best beekeeping management practices for the pollination, for the honeybees during pollination. One of those is the guide that's been put out by the Almond Board of California. Another website that has excellent information is the Canadian Pollinator Initiative at the University of Guelph. And honeybee pest management practices for California for almonds, I'm going to stick with those because they're very well done, and there's a guide here, and there are about 150 copies out in the hallway, so if you can't, do pick up one. And it really explains about why everybody should care about pollinators. So not just the beekeepers, right? Really the growers. And I've talked to several grower groups, and they do really care. They know that honeybees are essential for the pollination, for the fruit nuts that they grow. So they really do care, and it's really reassuring to see that. And let's see. So other than this comprehensive guide that the Almond Board has put out, they have these short guides that you can use, and it's laminated, so it's really nice. You can have it as a quick reference, and you can give it to your pesticide applicator or driver. They can actually keep it in their vehicle and refer to it quickly as they need to. So pick up one on your way out. I think it's very useful for everybody. And the biggest message here probably in this guide is communication. Everybody needs to communicate. Everybody needs to be on the same page. People need to be aware of what is going on. And when I'm talking about that, for example, they recommend to have contracts that actually include a pesticide plan for the growers in which pest control materials will be used during the time when the honeybees are out there in pollination in the orchards. And if the treatment is deemed necessary, then you should request from the grower to basically contact to have the grower contact you, or have them contact the county ag commissioner office so the beekeeper can be contacted and notified 48 hours in advance so they can do something with those colonies, move them if necessary. And I should also mention that you should, as a beekeeper, register the colonies, because this is also increasing the chance of you being notified if there are any pesticides applications going on around your hives, and you take care of your hives as you can. Another biggie, and I've heard this from Eric and of course others, and it's honeybees and insecticides. What's a honeybee? It's an insect, right? So you would expect that something as an insecticide might have an effect on an insect, a honeybee. So one of the best management practices here is avoid application of insecticides during bloom when the honeybees are present. This is also has an implication for the native pollinators as well, or native bees as well. We know, I think last year, bee losses have occurred during tank mixing, so the birds were trying to save time, honey, water, whatever, would mix fungicides and insecticides together. So the big recommendation now is to try and avoid mixing these two together and then applying them. And then a biggie are the insect growth regulators, which are, of course, insecticides. Currently, the most bee label warnings are based off of the effects on adults. EPA, I think, is working towards that, figuring out a different strategy for actually testing the pesticides and how they affect not only adults but other individuals in the hive as well. So hopefully that will come through, but definitely be wary of that and potential impact on not the adults only, but the immature bees in the hive as well. And there's some good information available if you're a grower or beekeeper and just want to familiarize yourself at the IPM UC Davis website. And then honeybees and fungicides. We used to think, oh, fungicides and bees, in this case, bees are not fungus, right? So fungicides would probably be okay, but we are not quite certain how all of the fungicides might affect bees. As I mentioned already, there have been some work showing that if the bees are exposed to fungicides, they actually might have an increased infection by nosema, so it could potentially be harmful to the honeybees as well. And one of the recommendations are basically to avoid exposing the pollen that the bees are foraging on to these spraying materials. So the recommendation would be to treat when the honeybees are not present or foraging. So for example, treat when the honeybees are not flying, so in the evening. Of course, you want to make sure that you're not spraying too late so there's enough time for these fungicides to dry off so the bees don't take it back home or they don't get exposed to them that way. And then one thing, this is sort of a given, don't spray the bees directly. They can't, even if it doesn't kill them, they really can't fly home right away if their wings are wet, right? So you want to avoid spraying the bees, basically. And then this is also another thing, if at all possible, report pesticide bee kills to the county ag commissioner's office. And people, I don't know why people always like, oh, I'm not reporting anything to the government, right? But they're not trying to keep track of anybody else. They're not trying to get you in trouble. We really need this data to make more informed decisions. It is beneficial for everybody, really. Because this is basically citizen science, right? The more data we have, the better we're able to support the honeybees and other pollinators as well. So if you can, I would highly recommend you report the pesticide bee kills, and the counties should come out and actually investigate that. Now, pest management practices for everyone. It is actually pretty simple, right? I took this out of the Xerces Society, the 3 steps for helping pollinators, so not just the honeybees. Again, you've heard this throughout the whole symposium so far. Forage is crucial. Forage is really important. I think Mark Winston was saying at one point, it's like you going to the grocery store and having only chicken available to you for a month. Chicken is great, right? Well, protein's good for you, but that's all you're eating for a month. Then the next time you go into the store, you only have, avocados available to you for a month. Again, good, healthy food, but it might not be the best for your own health to eat just one thing at a time, right? So they really need this variety of food sources, especially when it comes to pollen. And you've seen from 80 stuff as well, it can be very beneficial. So if you can, if you're not a beekeeper, if you're not planning on keeping bees, you're a homeowner, you have a backyard, even if you're in a city, right, you have a window that you could put a plant into, right? Try and plant a variety of blooming plants in your yard. And Michael has already mentioned something like, Michael has already talked about the Habitats Beehaven garden, right? We'll all be going there later, I hope, a lot of you. You'll be able to see some of the plants that are attractive to pollinators that are actually native to California that are drought resistant or drought tolerant. So there are quite a bit of options out there for you guys. And then protect nests and egg laying sites. Again, this is not for honeybees, this is more specific to native pollinators. A lot of them are nesting in the soil. So I think Marla mentioned don't put mulch, right, to make it available for them to be able to nest. And then if you can, again, avoid use of pesticides. If not, then definitely please follow the label instructions. And Amina is standing there threateningly, which I'm only assuming means, oh no, Michael will be standing threateningly in 2 minutes. So I will just briefly go over some of the work that we are planning on doing or are currently doing in my lab. Our mission, the mission of our program, the extension program, is to support the California stakeholders, no matter how big or small they are, through conducting basic and applied research and, of course, disseminating knowledge of honeybees and contemporary research findings through extension and outreach. But the first thing I want to cover is just quickly go through some of the research that we're doing. As I mentioned, I'm really interested in studying honeybee reproduction, not only because it's really interesting, but it can be applied practically. Queen is the only reproductive female in the hive, right? So anything that could negatively affect her could potentially negatively affect the entire colony. So beekeepers have been reporting the queen failures one of the top reasons of colony loss. We really want to, my goal is to really see what might be causing this and also how we can best support the queen health and quality, and then hopefully be able to support some more practical things like breeding protocols. So this is a fancy slide, fancier slide, but it's a really sad slide. This is basically a slide of probably about 7 or 8 years of work. So I always look at it like, well, this is great, but it's also, man, I could put everything on one slide. So I worked really hard for that, so I did. The main goal of my research was really understanding, so the queens undergo many changes after they mate, right? The main goal was really to understand what causes these changes. More specifically, how are these changes manifested in the queens that are in their final laying stage, in their final stage of sort of development, right? They're in this laying stage, they're all laying. Depending on how well they mated, now are they looking the same? Is it really just, okay, they're egg laying, they're excellent, that's cool, or even if you're egg laying, there are some differences between those queens? And what we basically found, yes, there are differences depending on what the queen has been, in this case, instrumentally inseminated with. And more importantly, that can actually have an effect on the colony, potentially negative effect on the colony. So it's not only that the queen is mated, but she needs to be well mated and mated with really really quality drones, right? So we'll be continuing work on this in my lab. We're now working towards identifying specific factors, so seminal fluid components such as proteins coming from drones. What are those specific factors that might be affecting specific post-mating changes? So if there's one protein, for example, that can affect and support the longevity of the queen, wouldn't that be nice? And also, a lot of these seminal proteins are supporting sperm survival. Brian has already talked a little bit about the queen failure and how they don't last as long. One of the things might be that they're actually, the sperm is losing viability while in the spermatheca, so we would like to see what is going on there. And again, this is all in hopes of helping to improve reproductive health of the queen. Of course, we're going to be working on some of the other factors that might be affecting the queen quality. I've already done some work on viral infection and collaborated on looking at the pesticide exposure and how that might be affecting the queen pheromone production, because the queen pheromone regulates the social organization of the hive. Interestingly, we didn't find any effect of the viral infection of the pesticides on the adult queens, so it didn't seem to change their pheromone production. However, student, Liz Walsh at Texas A&M, she was looking at the exposure of queens during development to pesticides, and she's actually found effects on pheromone production. So we're hoping to do something similar, or planning on doing something similar. I have a visiting scientist from, a visiting student from the Netherlands, Stephanie. Stephanie's here, you can ask her more about it. She will be looking at the effects of sort of a synergism between amitraz, so that's the miticide that's been being used for varroa control, and other agrochemicals in the hive, and how that might affect the development and reproductive health of the queens. My student Cameron Jasper, he'll be looking at how this synergism might affect the adult worker longevity. And we're all doing this in hopes to inform the management practices. So for example, how far in advance would you need to treat for varroa before you go out for pollination, before you're starting to see these negative effects? In addition to that, we are going to be collaborating with IR4 Project to look at some of the efficacy and honeybee safety of several novel biopesticides for varroa control. And then, as I mentioned, well, actually, I should mention, this is really the bulk of the lab's efforts, is to do extension and outreach. So we're doing a lot. I hope you think it looks like we're doing a lot, right? We're here to support the beekeepers, as I already mentioned, serving in various advisory boards, collaborating with farm advisors already, and then hoping to collaborate with various groups, so Project APSM, Almond Board, Beecorn Partnership, and members of those groups are all here, so hopefully you'll get a chance to meet them and talk to them more about what they're doing. And of course, we want to collaborate with the beekeepers. As Amy already talked about, beekeepers are a wonderful group to work with. It's a large group, so we're hoping to be able to set up some experiments, and this would provide huge sample sizes, I hope, which definitely would be very helpful. We're working towards developing web resources for the California beekeepers. California is a long state, or it's a huge state. It's a really long state too, right? So to be able to reach everybody, online materials are definitely crucial. We are providing various educational opportunities. We just put on a Queen short course. I've seen some familiar faces from that course here, so it's great to see you guys. I hope you had a good experience. You should talk to them, and we're going to be putting this class on next year, so hopefully I'll see some of you there. And we are, Michael already mentioned, we're in the process of developing the California Master Beekeeper Program. There are a few of them throughout the country. Ours is going to be the best. So, just letting you know. The way I see it is really, again, I'm hoping that the Master Beekeepers are going to be the bee ambassadors. I'm viewing them as the extension of our extension. And outreach. This is one of my favorite activities, really working with the kids. You guys are great, but the kids are awesome. They really get very excited about anything, and I like to point this group out because it's not always so straightforward. There are some challenges with this as well. This is a group of kids from the California School for the Blind. So when they first contacted me, I was like, oh my God, how am I going to do this? Obviously, I have the PowerPoint, right? I rely on visual aids quite a bit. But it turns out honeybees are a great system to do this with. Honeybees produce a lot of pheromones. A lot of these pheromones really smell wonderful. So the kids really enjoyed it. They like all the textures. They love holding drones because they're fuzzy. They don't sting. And then, of course, I took him out into the Holocaust honeybee haven garden, and they loved it. They could smell the flowers, they could touch the flowers. They also really enjoyed hearing the buzzing of the bees on those flowers. So it was really a nice tool for this, especially for this group of kids, to take him out into the garden. And I would, of course, like to acknowledge many, many, many people who have contributed to the research, the extension, the development of the program so far. And here's my information. If you have any questions, I think we might be out of time. So if you have any questions, let me know. Email is better. I've noticed that I have 2 minutes. Great. Or two questions. But I've learned that email is much better. For all the students out there, they tell you it's going to be starting off slowly when you get a job. That's not the case. So I have a lot of meetings, so email me, and I will get back to you. Okay, thank you so much. One question, it sounds like. I'll be around June, and I'll be at the Haagen-Dazs Honeybee Haven reception